This is Damiyanté and I will be answering question 1 to 3 regarding laboratory sampling and testing and transport and packaging of samples. The fecal parvo ELISA test is the most common test for parvovirus. ELISA stands for Enzyme-Linked Immunosorbent Assay. This test is performed in the veterinarian's office in less than 15 minutes. Canine Parvovirus PCR. This is the newest method of testing. This is where the DNA of the parvovirus is detected and amplified so as to be able to detect even small amounts of it. During a biopsy, parvovirus lesions in the gastrointestinal tract are of a classical appearance and are easily seen under the microscope. White cell count drop is a characteristic of parvovirus and can be seen on a blood panel when the infection is active and real. During sample collection, it is very important not to use tubes or swabs which have passed the expiry date. Always label tubes, suitably package samples, and always specify which test they are being sent in for. Samples are usually divided into two categories. Category A, which are infectious substances causing permanent disability or life-threatening disease to humans or animals, and Category B, which does not meet the inclusion of Category A. All samples must be packaged as follows. A primary container with a tube or vial containing the sample, usually made of glass, metal or plastic. It should have a leak-proof seal and should be labeled with a permanent marker. A secondary container, which is usually a watertight polyethylene box. To protect the primary container, it is usually supplied with cardboard or bubble wrap. And an outer container, which is usually a strengthened cardboard box protecting the secondary container. Fecal samples will be stored in a plastic or glass jar. Blood will be stored in a red top or purple top vial.
Hi, my name is Tasiya and I will be discussing the morphology of parvovirus. Thanks to the incredible technology we have today, viewing a virus is as simple as looking at it through a microscope. We've even discovered some viruses big enough to be seen through a light microscope. However, parvovirus, which is an unenveloped single-stranded DNA virus, is one of the smallest viruses, sitting with a diameter of about 18 to 28 nanometers. Thus, it can only be seen with the use of a transmission electron microscope. In order to view a virus with a microscope, it must first be stained. There are several methods of staining used, including positive staining and shadow casting, but the method generally used for parvovirus is called negative staining. In this method, a thin specimen is contrasted with an optically opaque fluid, which causes the background of the sample to be stained while the actual virus remains untouched. Immunostaining is another common method of staining parvovirus, in which an antibody-based method is used to detect specific proteins present in the virus, but this technique is used more for diagnostic purposes, whereas the negative staining is fundamental for confirmation of the morphology of the virus. Once the sample has been stained and viewed through the electron microscope, we see that it has an icosahedral structure that is formed by the proteins that make up the parvovirus. In the icosahedral structure, each side has 30 edges and 20 equilateral triangle faces with 5 meeting at each of its 12 vertices, which is pretty incredible considering the size of the organism. And that's the morphology of parvovirus.
Kenan Ganesan. Question 5. Parvovirus is considered a cellular contaminant and is unable to grow in standard cell cultures, which can make lab testing difficult. Question 6. Even though it cannot be grown in cell cultures, there are other methods to detect the virus. The SNAP Parvo test provides definitive results in just 8 minutes.
Hi, I'm Sejeng Matshidiso and I'll be answering the question, how will you determine that parvovirus is the pathogen causing diarrhea in dogs and not another virus or bacteria? Dogs infected with parvovirus show gastrointestinal signs. These include reduced or absent appetite, vomiting, and diarrhea. To detect that these signs are due to parvovirus, enzyme-linked immunosupplement assay or simply the ELISA test can be performed to detect parvovirus in a feces sample. This testing technique is a kind of immunoassay. A feces sample is added to the chamber and antibodies attached to parvovirus protein that may be available in the stool. A color-changing chemical is then added to the chamber. If parvoviruses have attached to the antibodies, the chemical will change color and indicate a positive test.
Explain the specific eclipse phase of the replication of this virus. Parvovirus is a non-enveloped, single-stranded DNA with an icosahedral capsid. The replication occurs within the nucleus. First, a complementary DNA is formed. It is then used as a template to form progeny virions. The virion penetrates into the cytoplasm via permeabilization of host endosomal membranes. Virion reaches nucleus where uncoating occurs. The single copy of DNA is initially converted to a duplex replication intermediate by cellular proteins, the non-structural replication proteins. This single-stranded DNA does not require special polymerases for replication. It must be noted that parvoviridae can replicate autonomously only in actively cycling cells. The eclipse phase lasts until new infectious virions are made, which is about 16 to 36 hours.
Hi, I'm Nomfundo. I'm here to describe the mutations that occur in the parvovirus of canines. Firstly, I'm going to describe what mutations are. Mutations are spontaneous and random errors in the copying of viral nucleic acid that can occur during the replication of viruses. According to different nucleic acid, the rate of mutation in the genome of DNA viruses ranges from 10 to minus 8 to 10 to minus 11 per incorporated nucleotide, whereas the mutation rate in the genome of RNA viruses is much higher, ranging from 10 to minus 3 to 10 to minus 4. Most mutations are lethal because the mutated virus has lost some vital information and can no longer replicate or compete with the wild-type virus. The first type of mutation I'm going to discuss is the point mutations. These mutations are the most common mutations of single nucleotide substitutes. Mutations may also involve deletions and insertion of single nucleotides or small or large blocks of nucleotides. The second mutation is the conditional lethal mutants that can grow under certain restrictive experimental conditions but can replicate under permissive conditions. The conditional lethal mutants are mostly common are those whose replication is blocked in certain host cells called host range mutants, or at certain defined temperatures called temperature sensitive mutants called adapted mutants. However, mutation results in a structural abnormal protein which, although may be functional at a permissive temperature, cannot maintain its structural integrity and functional conformation when the temperature is changed by a few degrees. And the last mutation is the attenuated mutants, which are many viral mutants caused by milder symptoms compared to the parental virus. These have a potential role in the vaccine development and also are useful tools in determining why the parental virus is harmful. Thank you.
Good evening, this is Daniel During and I'll be speaking about the transmission of the parvovirus. The parvovirus is extremely contagious. It spreads when an animal, in this case a dog, comes into oral contact with the parvovirus, which is spread by the feces of an infected dog. The resistant virus can remain on objects like food bowls or even floors for months and still transmit to dogs when oral contact is made with the virus. When ingested, the virus multiplies in the lymphoid tissue before moving to the bloodstream, after which it will start to attack rapidly dividing cells. The virus can also be transmitted by pregnant female dogs, which are infected to their fetus through the placenta. Parvovirus defense. For an immune response to occur against a viral infection, the host must first realize that there is a viral infection. It is this process that is antagonized by the viruses. This is possible because when a virus infection is sensed by the host cell, cytokines are produced that bind to proteins on the cell surface and trigger an immune response. But before this can happen, the viruses bind to the proteins on the cell surface, so the immune response is never triggered.